STARS crystallography lecture and workshop. So yeah, just quick introduction about ourselves, right? So what is STARS? You guys want to guess?
It's a tough acronym.
Okay, so what we do is we do grow so we do grow protein crystals and like DNA crystals, right? And so a little bit of background about ourselves. STARS is our full name is Structural Nucleic Acids Anti-Cancer Research Society. And so far we've worked on the structural part and like the crystal growth part and a little bit of the nucleic acid, but not yet the directly in the cancer part, but hopefully we'll get there soon. So STARS essentially is a nonprofit organization, and we're just the Atlanta branch of that nonprofit organization. So in the past like 3 or 4 years, we've hosted over like 8 or 9 different distinct events such as crystal growing competitions, summer camps, workshops, and all with the aim and goal of getting other students to understand what is research like, what is structural biology like, what is crystallography, why does it even matter? And we often do like these events, right, such as the crystal growing competition that we actually did with a Dodgen middle school class. The students had a lot of fun and they got awards for their good note taking and the beautiful crystals. And Professor Oyelere Group, who knows about the ribosome? Do you guys know what the ribosome is?
Yeah.
Okay, so what's a ribosome?
Responsible for translation of proteins from amino acids to proteins.
From amino acids. Very close, very close. Amino acids make up proteins.
Amino acids.
Yeah, so it connects them together. But where does it get the information from?
mRNA.
Yes, mRNA. Where does mRNA get its information from?
DNA.
DNA, that's correct. So that entire process is called the central dogma. So DNA gets transcribed into, or it's copied into RNA and then gets translated into proteins. So as part of the entire process of understanding what is the ribosome and how do they figure out this mechanism, Professor Oyelere and 2 other people, Venki Ramakrishnan and Professor Thomas Stites, these 3 people, they were awarded the Nobel Prize back in the early 2000s or so for the determination of ribosomes and understanding how does it create proteins. And so our work was pretty much inspired from her determination in sharing the information and love of science with other students as well. So here's an example of what a poster presentation looks like at the ACA conference or American Crystallographic Association conference. Conference happens each summer, and you just go in and give presentations. And so that's one of our goals for our student participants, give them a chance to go to the conference, present, and understand what it's like to give poster presentations. And so how did it start initially? Right. Initially, it was a high school crystal club, and we just like grew regular salt crystals and things like that. So that one is aluminum potassium sulfate. A lot of people give it the nickname of alum, right? And so you can do different creative experiments with it, right? And then we just built it off, and then got more students involved, like tried to outreach to them and show them what crystallography is like. And over these last 3 or so years, we've had over 350 plus students cumulatively participate in our programs. And so we're just really excited to share that type of excitement for research with all of you guys as well. And so ultimately, the nonprofit mission is to inspire all students in crystallography, bring up the next generation of student and scientific leaders through outreach and research. And then the mission, of course, is to engage and empower students in crystal growing, crystallography, and structural biology. And then ultimately, right, the question is, well, why does crystallography even matter in the first place? Like, it's like, okay, cool, bring up scientific leaders, yes, outreach, yes, research, very important. Why crystallography? Like, what's up with crystallography? And so for our collegiate branch, or our official name, the STARS Atlanta branch specifically, right? Even though, in the past we did crystal growing, right? The importance of sharing that love of the research aspect, of the wet lab aspect of crystallography is very important. That's why we hosted the first, crystallography workshop back in January of this year. So that was the first one. and then host a second one, and then a third one for the Walton High Schoolers, and then this is our fourth one. So thank you for coming, guys. Yeah, so we did that, right? And we had a lot of fun with the lecture, and all of our previous students attending and all of the lunch discussions, right? Our two guests. We also have some extra photos of preparing for the workshop in the later sections. So you guys will have a taste of what wet lab is like. And then, right, so it brings it back to the question, why does crystallography matter? Right, because in the past we were always doing inorganic crystals, regular sodium chloride crystals or like regular sugar crystals. Like, why do we want to grow DNA crystals? Why do we want to grow protein crystals? Right, so these are just examples of our protein crystals from the past. The goal is to find out, right, understand the structure, have the chance to do the research in growing these crystals, and then ultimately find a way to diffract the crystals, figure out the patterns, and then solve the structure. Why does the structure matter? Why do you need to find the structure of a crystal? Right, these are all questions we're going to answer through this presentation. So, right, for our focuses in the Atlanta branch specifically, this semester we're focusing on the computational side of things, like on the computer, how do you solve the structure? How do you figure out a good structure? And then the next semester we'll be focusing on how do you actually grow the proteins, how do you actually grow the DNA crystals, just like the previous pictures of crystals. And of course, we also have the American Crystallographic Association Conference. As part of our mission, right, we're trying to get students who are really interested in research and crystallography to get a chance to present at the conference. And because of that, the nonprofit is in the works of creating or the policy is already set up, but it's in the works of creating a fund for students or for STARS members or other people who want to join to be able to fund that trip to the ACA conference. And so I actually went to this conference this summer, and the next conference is they marketed it as their Diamond Anniversary Year conference. And so it's their 50th year next summer. And so if you're interested in something like that, definitely reach out and we can get you more information. So ultimately, right, the Atlanta branch vision mission is to create a wet lab research-based community for pre-health and pre-doctoral students, as well as foster student-faculty research collaborations on faculty crystallography projects, as well as looking into our own crystallography, protein, and nucleic acid projects. And the main reason why this is very important is because, think about it, like if you're on the like tech green, or like you're on one of those green spaces, what's something you almost always see? Can someone tell us? What's something you almost see? Tense what? Oh, tense? Okay. Are there any like scientists out there sometimes, or like students out there? What are they doing? Or like engineers? Do you see like drones flying around? Or like when they're working on their like aerospace projects, or like they're flying they're test running their planes, or like they're test running their equipment, or their robotics experiment. Like some of those engineering student researchers actively have their project out in the open. Like you can see it, and you can ask questions. What about the biochemistry and like biology and like the other engineering type of sciences? Like, where are those clubs? Those clubs, yeah. Yeah, in the lab. Except the fact is, the vast majority of the clubs, for example, ACS, ACS student organization club, the vast majority of those only focus on the social aspects of club making. So they ask a bunch of like professors to come and give talks. They ask a bunch of students to come and have lunch socials and all of this really good stuff. Resume building sessions, yes. But what about the chance to actually do the wet lab experiments? What about the chances? So the problem is that there are high barriers to this types of wet lab science. There's barriers because of mentorship barriers. You need a PI or a graduate mentor. There's time barriers. They don't always have the time to, for you to just ask questions. There's materials barriers and lab space barriers. Like, where are you going to get your equipment? How can you just run your experiment on your own? it's a good thing about crystallography is you can tackle all these things because you don't have to have like all too much like serious background research experience. You only need to know how to microcut, and then you can set up your plates, and then you see beautiful protein crystals, and you can get real research data out of that without, having to run like crazy organic chemistry reactions, or without having to work with hydrochloric acid or strong bases and stuff. So one of our aims is to provide wet lab research experience for students who are really interested in the like you can very easily ease into it without any like hard strings attached. And so that's one of our goals. Provide a mission with the opportunity, right, to grow these macromolecular crystals, protein, DNA crystals, and then learn about crystallography and have the chance to give a presentation at the conference. Any questions about this? Okay. Can you guys hear me in the back? Oh, that's not good. Okay, I'll speak louder. How about this? Okay, that's good. All right. Do you want me to repeat some of the information or? Okay, all right. So, yeah, and this, of course, is our high school branch. Our high school branch, right? They grow the inorganic crystals like copper sulfate, and they look into those types of aspects. We are aiming to have a third branch to join us, maybe next year, and so that's something we look into. And so crystallography, right, brings us back to the question: why crystallography? Why is it so important? Why does it even matter? Why do we care? So crystallography. Well, first, what is ography? What can you get out of crystallography? Yeah. Images, okay. Okay, graphs, yeah. What does the suffix ography mean? You takers. What does it mean? What does it mean? Do we know what ology means? Study of. Okay, then what about ography? Very, very close. Yeah, so ology and ography are actually very similar in their definitions. Both of them basically say the general study of the thing that was before the suffix. So yeah, crystallography is basically just the study of crystals. Study of crystals. And so what is crystallography? Study of crystals. There are three types of crystals: inorganic, organic, and macromolecular. So inorganic is something like your salt crystals or your geodes, or maybe like sodium chloride. Organic is anything that has a carbon molecule, a carbon atom in it. For example, sugar, but that's not necessarily like too crazy complex yet. And then macromolecular is something that's way larger, right? For example, if a sugar molecule was this big, then something that looks like a protein would probably be like this big, and then something that looks like DNA might be like super long, like this big. And so, and in comparison, an organic atom might be like this, like teeny tiny. And so yeah, macromolecular, there's 4 different types of macromolecules: DNA, protein, lipids, and also steroids. And you can grow these macromolecules just as how you can grow sugar rock candy. You can grow single crystals of proteins and DNA, and that's very important for research. So structure equals function. Do we see this? What is this?
Leaves.
Leaves. Yes. What is it doing? Okay. Yes. So it's taking in sunlight to do its chemical reaction, taking carbon dioxide, or taking carbon dioxide, convert it into sugar. Why are the leaves so broad? Exactly. So structure equals function. If you looked at that and it says structure dictates function, then what can you, so structure, they're so broad, right? The function to take in more sunlight because surface area. So just as how nature has figured out a way to perfectly match up the structure to the function, nature at the molecular level, the atomic level, also does the exact same thing. So for example, your DNA, right, it can just be all loose in the cell. So it has to be packed up nice and neat into a chromosome. So how does it get packed up into like these coils, these nucleosomes, and you have supercoils, and then those red circles, right? Those are histones. And so if you look closely at those histones, they have like these little amino acid residues like sticking out. Acetyl groups, yes. Do you want to talk about it?
Yeah, so histone acetylation is one of the two mechanisms that DNA is regulated. So we have histone acetylation because the DNA needs to be packed up tightly. The DNA molecules have these little tiny acetyl groups on the end, and they come together. And when we attach an acetyl to the molecule, it makes it bind tightly together and wrap around the histone and coil itself up.
Exactly. And so because you have those like that's how you have like histone deacetylases, histone or no, so HDACs and HATs to transfer those acetyl molecules, right, onto, for example, lysines to determine if there's charged or not charged. Because if they're charged, then they'll interact with the DNA, hold them close. But then if they're not charged, then they'll let go of the DNA. And so depending on like that on and off switch, you can figure out the function, right? So if you look at the structure, get to the function. So the big problem is, yeah, while you can see the leaf with your naked eye, can you really see all the supercoils under a microscope? Can you? Unless you use an electron microscope, but also electrons are very, very, very ionizing, right? So if you're constantly hitting your sample with electrons, it's possible that your sample might degrade. And so is there a feasible way to just straight up see your molecules to understand the structure and understand the function? Is there a way to do that? Yeah, crystallography is one of the ways you can do that. So one of the so the three main ways to look at those structures is through crystallography, solution NMR, and cryo-EM. But one of the most predominant ways has long been standing is crystallography, X-ray diffraction. And so yeah, what can structure tell you? It's like, okay, you got the structure, now what? Who knows what that is? Huh? Yes, it's written right there. Do you want to read it? Hemoglobin. What does it do?
That make up your red blood cells.
Yes, red blood cells. That is exactly correct. What do red blood cells do?
Carry on.
Exactly. So if the hemoglobin is what's making the red blood cells red, well, we shouldn't use that logic, but if you look at the hemoglobin, you see the iron. Well, you see the yellow, four yellow things in the middle of the hemoglobin. And so the four yellow things, each of them is represented by this structure. So you can say like, okay, this is a yellow thing. This yellow thing is a heat, right? And then that red tiny circle is the iron. And so if you take a look at that, what does iron like to do? Yeah, iron likes to oxidize. So iron really likes oxygen. So the iron in the heme likes to stick, or it likes to attract oxygen to the iron, which basically attracts oxygen into the hemoglobin. And then the hemoglobin thus carries the oxygen in the red blood cells to the different parts of your body that need the oxygen. And then they dump the oxygen and then take in the carbon dioxide and then dump the carbon dioxide in your lungs. And then you exhale and then you release the carbon dioxide. So by looking at the structure, you can be like, okay, well, this hemoglobin, right? Say, for example, if we have this hemoglobin protein, we have no idea what it does. And then we take a look at the structure, and we're like, okay, so it has these things, it has these characteristics, and these characteristics can help us look into what it does. And so specifically, right, this is what hemoglobin looks like. So this is a cartoon representation. This is what it looks like in a computer like a computer program. Specifically, see if this is, yeah, so specifically, this computer program is called Pymol. Do we know why it's called Pymol? It uses Python. Yes, that's correct. There's another program called JML. Do you guys want to guess why it's called JML? Yeah, Java. Java. Yeah, so that's from Primal. And you see all those different chains and amino acid residues, and that's just the one singular protein. And you can like go into the program, like rotate it around, zoom in, zoom out. Guess what? 50 years ago, they had to build the entire structure by hand. I saw that. I was like, that is crazy. This guy is crazy. I mean, he is crazy because he did win the Nobel Prize with his colleague in solving the structure of hemoglobin, and his colleague solving the structure of myoglobin, hemoglobin's cousin. So those two were awarded the Nobel Prize like mid 1960s or so. Yes. Well, yeah, so they had the technology to determine the positions of the atoms, but they didn't have the software to see it on, like, for example, a computer. Yeah.
It's kind of like the jigsaw.
Sort of, yeah, but just old-fashioned, I guess. Yes, very old-fashioned. When I saw that photo, I was like, wow, that is some dedication. So Max Proust, right? How did he get his PhD? Well, who in here is going to academia? Or plans on going to? Do you guys know? You guys, okay. So general synopsis, academia is basically going into research, like for example, going through undergrad, maybe getting a PhD, and then say, okay, I'm going to become a professor, or I'm going to become like a research scientist and help out with research. Going to industry is like, okay, I'm going to get my undergrad, maybe get a master's or a PhD, and then I'm going to work at a company and work on industry. And so he was interested in academia, and so he was looking for a research professor. So who in here knows what a PI is? Okay, so what's a PI?
The principal investigator. So they're the one who actually runs the lab, and they're responsible for getting grant funding and hiring undergrads, grad students, assistants.
Exactly. So for example, Georgia Tech might have a good number of research lab groups, and each of their groups will be like, for example, Oyelere Group or Lieberman Group. Yes. Okay. So for example, he's interested in academia. Academia is basically for research labs. And right, a PI is a principal investigator, and a principal investigator is basically the head of the research group. And he sets the PI sets the projects, the PI receives the grants, the PI assigns projects to students. And right, so Dr. Max Proust, well, at the time, he didn't have his PhD, but he was interested in some research. He was interested in getting his PhD. And so he went around, trying to find like a research advisor or a PI. And so he went to the PI known as, so this is also another diagram. But yeah, he also went to a PI that was known as the Great Sage at the time. He was like, Great Sage, how do you solve the mysteries of life? And the Great Sage replied, well, mysteries of life lie within the structure of proteins. And the only way to solve the structure of proteins is through X-ray crystallography. And so that was the Great Sage's response, right? J.D. Burnell, Dr. Burnell. He was the original one who got the crystal structure, or got the first protein crystal of pepsin to crystallize out. And he was the one who envisioned like perhaps what a protein would look like. And when Max Proust finally solved the protein of like hemoglobin or some other protein that he worked on for like 10-something years, he was like, yeah, I don't know why this protein looks so ugly. And yeah, I guess if you look at the green structure, it does look so weird and like blobby, right? But ultimately, each of those parts have different functions and everything. So, right, key thing: what does structure tell you? It tells you the function, tells you what it does. So indeed, here are some applications, right? Who can guess what's going on here? Okay, so there's the COVID virus, and there's a spike protein, and then there's a zoom in on some very specific amino acids on the spike protein.
is one of the steps towards how they got the vaccine in less than a year as compared to a regular trial of a decade. I remember they said like normally how a vaccine works is that you inject get the more dead version of it.
Yeah.
Dead version of it into dead version of the reason but this trial is risky and it often takes like a decade. So when the coronavirus emerged, they used some new technology where they isolated RNA from they isolated a small portion of the virus and instead injected that part. So now the body is like, this is the protein that we have to respond to in case anything happens.
Very good point. So just to repeat what she mentioned that is this perhaps what happened for the regular traditional vaccine versus your mRNA vaccine. And so the regular traditional vaccine, as you mentioned, is like basically you have a like attenuated or like a virus, weakened virus particle that had already half died or maybe has already completely died. But the process is long and arduous. Versus mRNA vaccine is you code for the spike protein and then your cells produce the spike protein for you, and the cells are like, wait, what is that? Even though it just produced it. And then starts creating antigens and antibodies to target the spike protein, and then train the body to say, if you see the spike protein again, then make sure to attack this. But yeah, that is definitely a very good point. And so, for example, right, those key amino acids most likely are amino acids that have very important significance in terms of structure and function of the spike protein and how, perhaps the body recognizes it as a like a marker, right, for the code for coronavirus. Then here's another example, right? CRISPR-Cas9, the genetic scissors. Jennifer Doudna and Manuel Charpentier were able to work on crystallizing it, and then also work on determining its structure. And then after they finally got a really good diffracting crystal of the CRISPR-Cas9, then they were able to fully get the full picture and then develop the genetic scissors. And the importance of this slide is basically to provide two examples of where crystallography was used to pinpoint the exact atomic resolution model of these proteins and then thus determine the function. Because the disadvantage of using the other two methods, cryo-EM and solution NMR, it can provide you with good resolution, but it only works for small systems. And then cryo-EM can only work for large systems, but has very low resolution. So overall, crystallography is the most widely used. In fact, it's so important that there has been these are a couple of Nobel Prize laureates that were awarded the Nobel Prize for their research related to crystallography. For example, Maurice Wilkinson, whose student was a student who got the first Rosalind Franklin. Maurice Wilkinson, his student was Rosalind Franklin, and then we got Francis Crick, James Watson, and then all the way down the line, right? And then we also have the two Bragg, the father and son duo, who also got Nobel Prize. And so this is only around 20 or so, or like 10 or so Nobel Prize laureates displayed. In total, there has been over 40 Nobel Prize winners for their work related to crystallography. And so crystallography is a very important technique. It can be used widely for different types of research and for determining, pushing the frontier for structural biology. I know this is a lot of examples where men have accepted the Nobel Prize laureate. But more recently, in 2019, we actually had the first female duo to accept the Nobel Prize, and that was Jennifer Doudna and Manuel Charpentier for CRISPR-Cas9. So yeah, crystallography, ultimately the importance is to determine protein structures, observe the natural world, and then harness that natural mechanism to create therapeutic drugs, for example, through CRISPR-Cas9. And so brings us to the club and research. So this is like, what is crystallography and what it does, why it's important, and real life examples. So club and research. This is going to be a little bit more background information about how the high school club originally developed. And even though it's about the high school, you can still take the information and then apply it to, for example, your own aspirations or your own club ideas. For example, if you're interested in creating like a neuroscience club or like a robotics club, you can also sort of think about your ideas while I'm going through these ideas as well. Because even though this was in the high school situation, the ideas for starting your own initiative and then sticking to it are largely applicable to any type of initiative you start with. So my initiative back in high school was to start a club. Why? Because I went to I had an opportunity to give like a presentation at a conference, and I was like, wow, this is so cool. Because originally other people were like, oh, I don't know what crystallography is. I don't know what that is. And I went there, and then everyone was talking about like crystallography, structural biology. People were growing crystals in outer space. They had all of their instrumentation, software, like machines, technology displayed at their exhibition hall. And then I saw that, I was like, wow, there's an entire crystallography world out there. And so I was like, yeah, I should bring this back to the high school and share it with my friends because I think this is really cool. So the initiative usually starts with probably an event or like some initial passion or drive. And so I started up the club, right? And even though we only had the chance to grow inorganic crystals such as sodium chloride or alum crystals, it's still a good opportunity to start experimenting how you want to take on your initiative. Right. And so we started experimenting and then sharing our passion for crystallography with other students and other high school students. And then we actually went to a teacher summer conference to share with the teachers why is crystallography important. How can teachers apply crystallography to their own classrooms? And so while you're understanding your initiative, understanding how you can tackle your initiative, you want to think about who's your audience, who are you trying to reach, what are the avenues you can reach them, and what do you aim to do? So for our high school branch, it was to share with others the love of crystallography, tell teachers how cool it is and how it could be actually applicable in the classroom, and then just be excited for science and share the love and passion of science with other students. And so why was I doing these things? Partially because the initiative, right? It starts with the initiative. And that initiative could be crystallography, robotics, neuroscience, as long as what you want to do starts with initiative. And the other reason, of course, was research. And I was in high school, right? And everyone was doing like internships. They were like doing this crazy stuff and that crazy stuff and this web lab opportunity and that web lab opportunity. And then I was like, I really want to get into that. And then COVID struck, and I was like, yeah, no, there's no way I'm going to be able to go to wet lab now that everything's shut down. And so because of that, I did the next best thing. Start up the club and then also look into some of the computer stuff that I could actually access on my own. So some of that computer stuff was this. Basically looking at one of the COVID-19 proteins that's important. This is, I believe, MPRO. This is MPRO. So I was looking at the protein that was important for targeting. And then I was also looking at the small molecules, right, that have already been FDA approved and trying to see if the small molecules, the FDA approved molecules, could perhaps bind with the MPRO protein. Why was I looking at FDA approved molecules? Because if it has already been FDA approved, then generally speaking, you could repurpose that for another protein target without having to go through additional clinical trials. So it saves you time. And so I did that for science fair project. And I looked into more research or tried to look into more research and join a couple of classes to try to, understand how do you read a paper, right? What is it when you try to write a research report? And then when I did AP research, I tried to tackle Cas12 because it was important for, supportive for diagnostics for COVID-19. And then Cas12 was like important protein that could be used in the diagnostic. And I was trying to see if there was some computer way or computer model simulation to make sure that, to see if that would be possible, to see the possibilities. And so a lot of the initiatives and club-based stuff and like your own projects can be largely, you know, like not self-driven, like you can initiate it on your own, right? You can try things out. That's what high school and that's what undergrad is like. A space to try things out, right? Like our Atlanta branch was only started up in the spring semester, and that's because we had heard about the opportunity that we could use one of the Bog's open biology labs for doing our own experiments. So if we hadn't heard about that opportunity and hadn't asked about it, then we might have not have had the chance to grow protein crystals or have workshops like these. So asking around, having your initiative, asking around, know where you want to go and know the audience you want to target. So ultimately, club and research, right? From my experience, I learned how to lead my students and help inspire others. And for the research part, learn how to read papers, synthesize ideas, write reports, and give talks. And even though this was back in high school, a lot of the stuff I learned was very applicable to, you know, these years in my undergrad years. And so, yeah, I just wanted to share that with you guys, just in case you guys had any initiative ideas, or in case that you guys, you know, want to look into research. So specifically about research and crystallography. Okay, so apparently this is the time for real. The first time that so why I say this is the first time for real, when I was in high school, you know, I never got the chance to get into wet lab, but this time I got to undergrad and had a chance to get into a wet lab for real. So how many guys want to apply for research positions? Okay, raise a high and okay, that's a good number. That's good. Okay, that's good. So here are some examples for what I did when I applied for research positions at Georgia Tech. So the thing about the chemistry and biology is the school usually sends these periodic emails just at the beginning of the fall or spring semester, maybe just 3 or so at a time, about open research positions available. And so one of them was at the CC lab, and I was really interested in what they were doing, and so I created my application, and then I sent it out. So I want to draw your attention specifically to the wording I used. I said, I think what's amazing about broadband current anti-Stokes Raman scattering, BCARS microscopy, is that it can induce many more inelastically scattered photons from molecules vibrating in space. And you don't have to worry about the jargon because half the time I also didn't really know what the jargon was about. For multitudes of vibration modes while enhancing detection of weaker anti-Stokes signals all at the same time. Thus, BCARS has, by magnitudes compared to other microscopy methods, much lower detection times, increased versatility, and increased detection efficacy. I like to work at CC Lab on streamlined cancer detection, since BCARS, first introduced by CC Lab, is such an invaluable technology in the field, and I want to be part of the project, part of the action, contribute to the machine learning of cancer patterns through my preparation of metabolites and my usage of such vinegar microscopy for fighting cancer. I'll be pursuing a biochemistry PhD. To work at the CC Lab will be invaluable, preparing intellectually and academically for my future research endeavors. So what's your take, guys, on this so far? Like, what's the tone? Or like, what is it? Imagine if you were a PI and you read that. What do you guys think? That's a what? Okay, so she said that's a student I want in my lab.
They actually did research and they might actually be interested.
Okay, they have a little bit of research experience and might be interested. Okay, so we can just sort of keep this in mind. Generally, the tone is like excited, sort of doesn't know exactly what BCARS can completely do, but definitely very interested. This is another one. Dear Professor Williams, good morning. My name is Susanna Huang. I'll be a first-year biochemistry major at Georgia Tech this semester. I was looking at some of your research papers and lectures, and I find that your ongoing research projects on tracing the origins of the pre-ribosome to be especially exciting. In fact, I'm very enthusiastic about this research. Would like to seek a post for any available position in your lab this fall semester. If you have the time, I'll be happy to meet with you and discuss with you how to contribute to your lab. I'm generally available all this week. Please find my resume attached below. How's that one? You think that one's better? Why?
software, but you're you actually put the time to introduce yourself as who you are. Why would you like this?
Mhm.
Because if I were to be the first mail without knowing where your research, I would have probably assumed that you're sending me an article.
Exactly. Like, because think about it. The B-Cars microscopy is like what the 60-minute lab perfect. Like, this is what they do. This is their job. Like, they know exactly, what the B-Cars is about. They don't need to hear me repeat what they already know. It's like as if you're a teacher that's teaching multiplication. You're like, hey, student, or the student comes to you asking you, actually, that's a very not so good example. But it's like the student telling you something you already 100%, 200% already know. And it's like, wait, what? So this one, right, more enthusiastic, I don't know, more enthusiastic. Let's take a look at this one. I like to work at the Oyelere lab on optimizing and synthesizing the 3-HPT inhibitor because for me, this type of research, small molecule drug discovery, is and always will be one of my most favorite research areas. So far, I've performed exciting GRUMACs, molecular dynamic simulations for CRISPR-Cas12 predictions. I've also conducted molecular docking simulations, screening for potential SARS-CoV-2 MPRO inhibitors. Performing experiments for drug discovery is what I live for. It's exciting, audacious, and meaningful. Not only will I satisfy my conscious thirst for knowledge, but also I will simultaneously contribute to the betterment of society. Though I do not yet fulfill the Chem 2311 course requirement, I am a very motivated and passionate learner, and I can learn new concepts very quickly and easily apply them to the synthesis experiments in the lab, especially when I'm invested in synthesizing the inhibitor I would have optimized myself, post-graduation PhD. So thoughts? Way too excited? Okay. Overexcited. Any thoughts? Yes.
College essay, like application essay, like there's a lot of there's definitely all the stuff like kind of this thing over it's very exciting part, but it's focusing on the super exciting and contribute to the better society. That doesn't feel like that needs to be in this email.
Okay. Very, very good point of view. So I guess what I wanted to bring attention to was the fact that I had previous related research experience that I legitimately looked into. That is legitimately connected to the research of the Oyelere lab. And because of that, I know what I'm talking about. And I know what I would be contributing if I did join the Oyelere lab. And so this situation is very different from the first situation, because the first situation was like, I sort of know what the B-Cars thing was about, but I didn't actually know how it'll be used. But here I knew what therapeutic drug design was. I knew what docking was. I knew what it means to synthesize molecules. I know what it means to dock molecules. I knew the overall idea already. And so focusing that, or like amplifying that part and talking about my qualifications for the position is important. Right. And this is applicable to when you guys, if you guys want to find research positions, look into research that you may or may not have already seen before and amplify the parts of your experience that are directly applicable to the research possibility to show or demonstrate your capabilities and your qualifications. So, yeah. So that circles back, right? Because the docking, it has to dock on something, right? And that something is protein. Where did you get that protein from? Crystal structures. So it's very important. Crystal structures, crystallography is important for determining the function, also for designing the therapeutics as well. And so I know there's like maybe 6 or 7 minutes left. I'm just going to very briefly go through what it was like first joining. So just joined, right? And then started doing some synthesis experiments. And at first it was a little bit rough learning everything, but after I started figuring things out and had the help of the lab members, I started to enjoy it. And doing TLC, looking at spectra, right? And this was just an example of one of the presentations I gave. I'm not going to specifically go through it this time because there's not enough time, but just general idea, prostate cancer is one of the leading causes of cancer-related deaths in men. And one of the most effective ways to treat this cancer is to either reduce, so there's a treatment called androgen deprivation therapy, and what it is, you would reduce or eliminate entirely the androgens, and thus you can cancel out the cancer growth. Unfortunately, sometimes the androgen receptor gets mutated, and even in the absence of androgens, the cancer growth still happens. And so how can we deal with this? This is called castration-resistant prostate cancer. It's not affected by androgen deprivation therapy, the original gold standard that usually works, but in this case it doesn't work. So in these cases, what do we do? In these cases, what we can do is directly target the androgen receptor upstream. So you can just deactivate the HX6, deactivate the HSP90, and that'll just automatically deactivate everything else. You can deactivate it with an inhibitor. And so this HX6, because of the pathway, is one key target that can be used to treat this type of cancer. And that's what the Oyelere lab is very interested in doing, finding inhibitors to target the HX6. So that's 3-HPT. And so, just the big problem about 3-HPT is that it's very polar, right? So it can't enter the cells, even though it works perfectly fine against the HX6. A very little tiny quantity will perfectly deactivate the HX6 almost entirely. But if you try to put it into the cancer cells, right, it can't get in because it's polar. It can't pass through the nonpolar membrane. It's an idea as well. What if you just added some more nonpolar characteristics? Could you just sneak it through and then target the HX6? So that is what the project was about, making those alkyl versions and then sneaking it through. And so I did the docking, right? And so these are just in the computational side of things. I have the protein, I have my compound, and then I docked it. And I was like, does this seem better or does it seem worse? And if it seems better, then we might just go into the lab and synthesize or make the physical compound. And so the rest of the project is just making the physical compound. You do a series of chemical reactions, alkylation, thylation, demethylation, and then troubleshooting, right? And this is just reactions in the NMR, right? So, for example, this is just example. So the NMRs show the hydrogen peaks. So you can tell if your compound was synthesized or not based on the hydrogen peaks. Like, oh, I know this hydrogen peak should be here, and so I should expect it right here. And so you'll be able to tell if your compound was synthesized correctly if you have those peaks. And so ultimate idea, right, from the project was I was able to synthesize a good number of compounds. And the next steps, of course, to synthesize more. And right, you have acknowledgement. This is an example of what a presentation can look like. And so you put your acknowledgements, right? Professor Oyelere and mentor Ryan Kern. That's actually one of the guests who will actually be here. So you can ask him questions if you'd like. And fellow Oyelere Group members. And so, right, to bring the message home, I did the docking analysis. Where did we get those crystal structures? Where did we get those proteins from? We got it from crystal structures. Otherwise, how would we have known, like, oh, how do you design this compound if you don't even know what the protein looks like? If you don't even know what the active site looks like? How do you design the compound? If you can't even see how do you solve a problem if you don't even know what the problem looks like? Right? And so that's the importance of crystallography, of seeing what your problem looks like, seeing what the protein looks like, or seeing whatever drug anti-cancer target looks like. And then based on that, you can design your molecule, and then you can go in and fit it in, and then see if it inhibits your protein. Right, and the STARS organization, over the summer, I had the chance to go intern at the Berkeley lab, and I grew some protein crystals and some DNA crystals, and I have some lysozyme crystals there as well. And so the idea is that when we have the second crystallography workshop in the fall semester of this year, we hope to be able to remotely connect to the Berkeley Lab synchrotrons so that we can display in live time what diffracting the crystals look like. So we have the crystals, right, and you shoot the X-rays through. What does the diffraction actually look like? How does the data collection really look like? So that's one of the ideas we hope to have in the fall semester of this year. Right, and so ultimately, through crystallography, you can get the structure of the protein, you can design therapeutics to target your protein, and also, while you're doing all that scientific stuff, you can still experience the sheer beauty of growing the crystals in the first place. So that is the crazy part about crystallography, is it's scientific, it's beautiful, and because of those two things, you can bring new students into the field because you can actually see the crystals, and there's huge therapeutic drug discovery applications related to this field. So ultimately, right, crystallography is very important in the natural world, therapeutic drug design. Based on your initiative, you can create your team, lead them, and then teach them. And you can also learn how to read research papers, give talks, write reports, and then bring people together, right, on your initiative. Then ultimately, perhaps join a research group with the type of research you want to do, become competent or even master it, and then bring forth your own perspective. Maybe join your team and your research group together on collaboration, or maybe start teaching your students so well that your research group or other research groups might be interested in having some of your students join their labs. It could be an advantageous way for both research groups and your organization or branch to collaborate with each other and network. So yeah, because who knows, research is very, very important. There's a lot of areas of research. Crystallography is one area that is at the intersection between chemistry, biochemistry, structural biology, medicine, biophysical sciences, computational science. And so it's very interdisciplinary, and that's why we want to create a network of students who are very interested in research and very interested in sharing research ideas and just networking with professors, and that way we can build up a group of students on campus who enjoy research and enjoy sharing the love of research with others. So yeah, thank you so much.